This procedure uses the Virochip, a pan-viral microarray assay, to analyze clinical samples for both known and unknown viruses. First, nucleic acids are extracted from clinical samples. Next, the extracted RNA is reverse transcribed using random primers, second-strand cDNA synthesis is performed, and the cDNA library is amplified with random PCR. Then, the amplified cDNA is labeled with fluorescent dyes and hybridized to a Virochip microarray. The final step is to scan and analyze the microarray. Ultimately, results can be obtained that show the presence or absence of a virus or several viruses through analysis of the Virochip microarray.
Broad-spectrum detection methods such as a Virochip microarray are needed in diagnostic microbiology. Because common clinical syndromes such as pneumonia are not specific to any individual pathogen, and because of the continual emerging threat of novel viral pathogens such as the SARS coronavirus and 2009 novel H1N1 influenza virus.
This method can help answer key questions in the field of diagnostic microbiology, such as what percentage of undiagnosed infections in the clinical setting may be caused by as yet unidentified novel viruses. The implications of this technique extend toward better diagnosis of acute clinical illness in the hospital, because we are testing for hundreds to thousands of potential pathogens simultaneously.
We first had the idea for this method in 2002, when we decided to apply the newly developed technology of microarrays, pioneered in the laboratory of Dr. Joseph DeRisi at UCSF, to the detection of